Cancer. It's undoubtedly one of the most complex global health challenges of our time. But what if nature has been hiding potential answers right in plain sight? Welcome to this explainer. Today we're going to objectively evaluate the biological mechanisms behind traditional medicinal fungi, specifically Cordyceps. We'll be looking directly at the rigorous science to figure out its actual potential role in oncology.
To do this, we're breaking our clinical deep dive into 6 parts. We'll meet the fungi, look at the cancer research and evidence, explore the mechanisms of action, and dive into the power of Cordycepin. Then, we'll cover the limitations and future directions before delivering a final scientific verdict.
Section 1. Meet the fungi. Because to truly grasp the science here, we have to start with the actual organisms we're talking about.
So, Cordyceps sinensis and Cordyceps militaris, these are entomopathogenic fungi. That's a fancy way of saying that out in nature, they actually grow as parasites on insects. It's wild. Now, they've been revered in traditional Asian medicine for centuries, but today, they're the subject of intense, modern laboratory analysis. Researchers aren't just looking at the whole mushroom anymore. They're actively isolating complex, highly potent bioactive compounds. We're talking about things like cordycepin, adenosine, and specific polysaccharides, all to see exactly how they interact with human cells.
Section 2. Cancer research and evidence. Let's look at how these isolated extracts actually behave when they encounter various forms of cancer.
The breadth of the current research is honestly remarkable. In lab models of non-small cell lung cancer, we're seeing suppressed proliferation. In breast cancer models, reduced metastasis. And in colon and leukemia models, we see significant cell cycle arrest and controlled cell death. But, and this is absolutely crucial for scientific objectivity, in androgen-responsive prostate cancer models, Cordyceps actually promoted tumor growth. It did this by stimulating testosterone pathways. So this critical contraindication really highlights that we're dealing with a complex biological agent here, not some kind of universal magic bullet.
But we aren't just relying on cell cultures and mouse models. We actually have human data now. A major 2024 meta-analysis evaluated exactly 928 lung cancer patients spread across 12 randomized controlled trials.
And the data is really compelling. It shows that using Cordyceps sinensis strictly as an adjuvant treatment, meaning it's used right alongside standard chemotherapy or radiotherapy, not only improved the overall tumor response rate, but it significantly reduced severe adverse reactions. Patients actually experienced lower rates of radiation pneumonitis and myelosuppression compared to the folks receiving standard therapy alone.
Section 3. Mechanisms of action. To really understand those clinical outcomes, we need to zoom in on the cellular battlefield. How exactly are these fungal compounds disrupting cancer survival pathways?
Well, research indicates the efficacy of Cordyceps comes down to a multi-target approach. It dynamically modulates 4 primary cellular mechanisms. Basically, it forces cancer cells to die, stops them from dividing, boosts the body's immune response, and prevents tumors from spreading. It's hitting the cancer from multiple angles. When we chart out the apoptosis pathway, we can see exactly how these extracts trigger a sort of domino effect inside the cancer cell. It starts by activating the cell's own DNA damage response. This then upregulates crucial tumor suppressor proteins like p53 and p21, and that eventually activates the cascade, which essentially forces the malignant cell into a controlled systemic self-destruction. It's a programmed death. And the science here is constantly evolving. Very recent data looking at lung cancer models revealed that optimized Cordyceps extracts can induce something called apoptosis. This is a highly specific iron-dependent form of cell death. What happens is, the extract causes iron to accumulate inside the cell. This leads to severe lipid peroxidation and suppresses a key protective enzyme called GPX4. Ultimately, it destroys the cell through overwhelming oxidative stress. Just look at how fast this happens. Tracking the disruption of breast cancer cells in a lab setting shows us how rapidly and systematically the fungal extracts alter the cell's internal environment. Within just 2 to 4 hours, a quick spike in antioxidant enzymes gives way to the early indicators of cell death. By the 16-hour mark, p53 activates and the mitochondria are under severe stress. And by 72 hours, we see profound morphological cell disruption. The cancer cell is quite literally dismantled from the inside out. Section 4. The power of Cordycepin. Central to all these impressive molecular interventions is one specific, highly potent compound in the Cordyceps arsenal. Meet Cordycepin. It's a purine nucleoside derivative, and what makes this so fascinating is its structural similarity to adenosine, which is a fundamental building block of our own DNA and RNA.
Because it looks so similar, it acts like a molecular Trojan horse. It infiltrates the tumor cell and completely disrupts critical RNA synthesis and signaling processes. It effectively cuts off the cancer cell's ability to communicate and replicate.
But how we get that compound matters immensely. Network pharmacology studies emphasize that extracting the fungi at exactly 80 degrees Celsius yields the highest concentration of cordycepin. This optimized temperature unlocks significantly more potent anti-cancer activity compared to other extraction methods.
The current scientific consensus is becoming really clear. Isolating and refining specific compounds, like cordycepin, rather than just consuming raw or crude extracts, holds incredible potential. As Gunter and colleagues noted in 2026, Cordycepin represents a highly promising chemotherapeutic lead. It really could pave the way for entirely novel drug development.
Section 5. Limitations and future directions. Now, despite all this overwhelming molecular evidence, we have to maintain our clinical objectivity and clearly address the actual boundaries of our current understanding.
While the laboratory data is incredibly robust, we have to remember that the vast majority of it comes from in vitro cell cultures and animal models. High-quality randomized human clinical trials remain severely limited right now. And because specific contraindications exist, like we saw earlier with prostate cancer, Cordyceps should absolutely never be considered a standalone replacement for standard oncology protocols. It absolutely requires strict medical oversight.
Looking forward, though, the research is pushing into some incredible territory. Scientists are now exploring precision oncology applications. For example, recent studies are utilizing targeted nanocarriers to deliver cordyceps extracts directly to cancer cells. They're also looking at how it synergizes with cutting-edge immune checkpoint inhibitors, how they can structurally modify cordycepin for drug discovery, and how to standardize artificial cultivation to really maximize those bioactive yields. Section 6. Final scientific verdict. Let's distill all of this source material into a definitive scientific conclusion. Throughout this explainer, we've seen that the evidence for cordyceps is complex, it's fascinating, and at the cellular level, it's pretty undeniable. It possesses biologically active compounds with profound multi-pathway anti-cancer potential in laboratory settings. It bridges this gap between traditional medicine and modern pharmacology that we are really only just beginning to safely cross. The laboratory promise is huge, for sure, but robust human clinical trials are absolutely imperative before any definitive therapeutic recommendations can be established.
So as we continue to investigate this microscopic warfare taking place between fungi and cancer cells in labs all around the world, we have to ask ourselves, could the intricate biochemistry of ancient fungal ecosystems actually hold the exact molecular blueprints required for the next era of precision oncology? Thanks for joining me for this explainer. Keep learning and keep asking the big questions.